Chapter 2. What is cytology? The branch of biology concerns with the structure and function of plants and animal cells. Cell is the basic unit of life. Cell, tissue, organ systems, and organisms. Important terms contents. Organelles. Organized of specialized structure within a living cells. Prokaryotic cells. Single cells organism with neither distinct nucleus with membranes nor other specialized structure. Eukaryotic cells, organism consisting of cells or cells in which the genetic material is DNA in the form of chromosomes contains with distinct nucleus.
The cytoplasmic membranes and cell wall, aka plasma membrane or cell membranes, thin structure that completely surround the cell and typically 7.5 or 8.0 micrometer thick structure fluid mosaic model. Function is to support and protect cell functions, defense against pathogen, and provide shape to plant cell.
The cell membrane function, regulating the movement of material into the cells. Selective permeability, give shape, strength, and protection to cells.
Yes, the genetic material of eukaryotic cell is found mainly in nucleus. Nucleus is the most important organelle, typically 5 micrometer in diameter.
Structure contains nuclear membranes, two-layer inner and outer layer. Out layer. Outer layer is continuous and connected with another organelle, the rod endoplasmic reticulums. Chromatin contains DNA and proteins. Nucleus region in which RNA are synthesized and aggregated.
Function of nucleus, regulating cell activities which are continuously happen by determining which protein molecule should be produced, involved in hereditary function, involved in cell division and located of ribosome synthesis, nucleolus.
Organelles. First is mitochondria. Second is plastids. Third one is endoplasmic reticulum. Fourth is ribosomes. The fifth one is the Golgi apparatus. Six lysosomes and seven vacuoles.
First, mitochondria exist in all eukaryotic cells. Higher in number of mitochondria, higher level of activity. Each one until 10 micrometer long. Move freely in cytoplasm with no defined shape.
Plasticids only exist in plant cell. Have three types: chloroplast, green color plastid, and chlorophyll. Chromoplast, orange, yellow, red color plastid. Example: flowers. Leucoplast, floris plastid, no pigment. Most important chloroplast, site of photosynthesis in green plants and green algae. Size 0.5 until 2 micrometer wide, 10 micrometer long. Shape vary. Location do not stay in fixed position. structure surrounded by outer and inner membrane.
Plasmic reticulum, largest structure in eukaryotic cell, made up from phospholipid bilayer, a system of sac which enclose a membranes known as cisternae. Connected to nuclear membranes, the Golgi apparatus and lysosome classified based on the present absence of ribosome, rafter, and smoother. Smooth ER, tabular or sac shape, smaller than rough ER. Function involved in synthesis and transposition of phospholipid, phospholipid, steroid, and fatty acid. Detoxification of hazardous chemical, pesticide, and sarni, san, carcinogen.
Routes ER, second vesicle with ribosomes on their surface. Function: temporary storage for protein synthesis by ribosomes, transporting protein from one part to others part in cell using systemal space.
Okay, the difference between smooth ER and rough ER: synthesis, lipid, and steroid, metabolize carbohydrate, the difference between smooth ER and rough ER: synthesize lipid and steroid, metabolize carbohydrate, detoxifies poison, store calcium, lysogen degradation site.
Routes ER has bound ribosomes secrete glycoproteins, distribute transport vesicle proteins surrounded by membranes, is a membrane factor for the cells. Some occur in all cells, especially the actively synthesizing protein. Consists of two subunits, small and large subunits, synthesized by nucleus free floating in cytoplasma, free ribosomes, and attract to route ER. Made from RNA and protein, function protein synthesis.
Okay, the Golgi apparatus occur in all animal and plant cells. Consists of a group of a membrane surrounded sac. Occur mostly in secreting cells. Function: processing, modifying, and packing new protein from ER into glycoprotein. Secretion of glycoprotein and formation of new membrane and cell wall.
Lysosomes only exist in animal cell. Spherical shape contains digestion enzymes from the Golgi apparatus. Intracellular desiccation or via photosynthesis, destroying bacteria and foreign substances in cells involved in autolysis. Lysosomes membrane will be dissolved and all the digestion enzymes will destroy the wall cells.
Okay. Vacuole. Vesicle filled with fluid and which is enclosed in a single layer membrane known as tonoplast. Sap filled with H2O, organic material such as glucose and amino acids. Mineral salt dissolves O2 and CO2. Small cells, small vacuole or have none of it. Plant cells have a large vacuole and increasing in size as the cell become more mature. Is to storage for waste material. Cell enlargement filled with sap will further increase the cell size.
Mitosis introduction. What is it? A type of cell division which involves the division of the nucleus to produce two daughter cells. Daughter cells contains same number and same kind of chromosomes as the parents, where all some somatic cells, all body cells except gametes.
Okay, somatic cell contain two set of chromosome. One set from female parents and another from male parents. Two set of chromosome. Diploid cell. Somatic cells. Cell with single set of chromosome. Haploid cell and gametes.
Prophase centrales move opposite pole. The chromosomes coil up, condense and shorten.
Let prophase. Each chromosome appear as two identical chromatids. Sister chromatids attached at centromeres. Nuclear membrane breaks down and nucleolus disappear. Formation of spindle fiber.
Metaphase contromers attached to spindle fibers. Chromosomes move to cells equator.
Centromere of each sister chromatid divide into two. Each will separate and move to opposite cell. Spindle fiber pulls the centromere towards each pole. When chromatids reach the pole, they will become independent chromosomes.
Telophase. Two sets of chromosomes at each pole. Chromosome discondense like structure. Spindle fibers disappear. Nucleus membrane begin to form around both sets of chromosomes. Nucleus reappears.
The result of mitosis. Two daughter nuclei are produced, each with same chromosome number as parent cell. 1. Pair homologous chromosome line up at the cell equators. Chromatids of each chromosome are attached to spindle fiber.
Anaphase 1. The pair homologous chromosomes separate from one another and pulled by spindle fibers. The sister chromatic remains attached.
Telophase 1. Chromosome reach the pole. Each pole has a haploid chromosome set and spindle fiber disappear. Nuclear membranes and nucleolus reappear and cytokinesis occur.
Prophase 2. Control the duplicates and move to opposite pole. Each chromosome consists of two sisters chromatids attached to centromere. Nucleus membrane and nucleus disappear. Spindle fiber formations.
Metaphase 2. Chromosome line up at the cell equators.
Anaphase 2. Sister chromatids separate to become independent. Spindle fibers pull chromatic to interphase, divide into three subphases. G1 phase. Cell grows by producing protein and cytoplasmic organelles. Cell growth and differentiation occur. S phase. Synthesis. Synthesis of DNA occurs. DNA replications. G2 phase. Cell growth by producing protein and cytoplasmic organelles. Cell growth and differentiation occur.
Content. In interphase, chromosome appear as threads-like structure chromatin. In general, interphase is where the cell prepare for cell division.
M phase. Once interphase complete, M phase begins, in which mitosis and cytokinesis occurs. Shortest part of the cell's cycle because interphase make up about 90% of the cell cycle.
There are four stages of mitosis. First, prophase, metaphase, anaphase, and telophase.